So now we get to talk about the human microbiome. At the beginning of this term, we talked about the golden age of microbiology and a lot of the researchers who were important in the golden age of microbiology. The golden age of microbiology was initiated with the advent of microscopes that were powerful enough to discover and explore the microbial world for the first time. A lot of researchers now believe that this age that we are in right now is the second golden age of microbiology because of the advent of next generation sequencing technologies. So next generation sequencing technologies started around early 2000s. And with the advent of next generation sequencing technologies, we've been able to explore the microbial world in a level of detail that was completely inaccessible to us before that. So before next generation sequencing technologies, we could sequence the DNA of microbes, but only when they were from a pure sample. And as we discussed many times earlier in this course, the majority of microbes cannot be isolated and cultured in the lab, which means that much of the microbial world was invisible to us. Additionally, what microbes do in isolation can be very different from what they do in a community. So how do microbial communities behave? What is the ecology of a microbial community? These are things we could not discover before. We could, put in a substrate, like maybe some organic material that was decomposing, and then look at what came out the other end for a particular microbial community. But we could, but who did what and how they did it, that was a complete black box until the advent of next generation sequencing technologies. This being the golden age, the second golden age of microbiology, means that a lot of what we're understanding about the human microbiome is still evolving. There's a lot that we still don't understand. Our understanding is changing every year, but this research being cutting edge, it's really mind-blowing, and the things that we're learning are really fascinating about how microbes work together in a human body or in a soil ecosystem or in the ocean ecosystems. So we're going to focus on the human microbiome as opposed to these other microbiomes that are also really fascinating out there. You may think of yourself as just a person, but you are not. You are a thriving ecosystem of microbes. There are 10 times more microbial cells living in and on your body than human cells, and there are over 100 times more microbial genes acting in and on your body than human genes. So because this is the end of the term, I want to incorporate some review for the final exam, which is cumulative. So I'm going to remind you what a gene is. A gene is a code for a protein. So why might a gene be an important measure of the relative effect of the microbial body versus your human body? Well, these proteins that are encoded by genes, they are the little machines that get everything done. So a microbial this means that there are perhaps hundreds of times more microbial proteins acting in and on your body than human proteins. And some of these microbial proteins, we know exactly what they do. We know that they are involved in digestion. We know that they're involved in producing vitamins for us or antimicrobial chemicals that keep us safe from disease. And some of them we're still learning what their importance are, whether they're important to our bodies or whether they're not. So the microbiome is the control center of our immune system. Microbial imbalance, which we call dysbiosis, has been implicated in many diseases. And here are some headlines from peer-reviewed research on the kinds of diseases that are now being implicated with dysbiosis. So allergies, diabetes, irritable bowel syndrome, dermatitis, obesity, and a variety of disorders involving brain and nerve function because of the effect of microbially produced compounds on mood, sleep, and cognition. So this is just a partial list of some of the diseases in which dysbiosis has been implicated. Now we know that creating overly sterile external and internal environments detracts ultimately from human health. To encourage a healthy microbiome, antibiotics should be prescribed and taken with extreme caution, and children should play in the dirt. A human microbiome researcher, Jonathan Eisen, says we should treat our microbiome like one of our organs. We need to treat it with respect. So now we will begin talking about how the microbiome affects our health and behavior. What are the mechanisms? Much of what we will talk about draws heavily upon what you've already learned about microbial biology, ecology, and especially human immune responses and microbial mechanisms of pathogenicity. So let's start with the gut, because that is the microbiome that is best studied so far, and it's also where most of the action lies. This is a link to a 6-minute video made by NPR, and I'm going to link to it on the course website. It's a really it has beautiful artistry. It's a beautifully done video that gives you an introduction on the kinds of research that's now being done and what we currently understand about how the microbiome affects our health.